It is my pleasure to introduce today's speaker, Dr. Stefania Carobbio, to our yellow webinar. She is group leader at the Centro de Investigacion Principe Felipe in Valencia, Spain. She earned her PhD in biology from the University of Geneva.
No, Geneva.
Geneva, yes, thank you. Following her PhD, she was awarded with prestigious fellowship from the Swiss National Science Foundation and the Marie Curie program, which enabled her to join the University of Cambridge, the Institute of Metabolic Science, and then at the Wellcome Sanger Institute, where she developed innovative stem cells models to study fat biology and energy metabolism. Since 2021, she has led her own research group supported by European and Spanish competitive grants, including an ERC project, the CDGEN funding, and National Ministry Awards. Her team focused on promoting healthy adipose tissue function, activating brown fat as a therapeutic approach for obesity, and more recently, investigating the impact of endocrine disrupting chemicals on metabolic associated steatotic liver disease. Please join me in welcoming Dr. Stefania Carobbio.
Okay, so should I start?
Yeah.
Okay, so thank you very much to the, first of all, thank you to the organizer for giving me the opportunity to present this webinar. And I will talk about this new European project that I'm part of, and that is just started 1.5 years ago, so I don't have many results to present, but still I want to introduce the project and what we are doing there. So the title of my talk is Effects of Endocrine Disrupting Chemicals on the MASLD Progression. So first of all, I want to introduce some concepts that are key for understanding the project. So first of all, the table of contents, sorry. So I will give a little introduction and put in the context the EDC MASLD project I'm part of, talk about the main hypothesis we have, give a little bit of an overview from the iteration on the PFAS, that are the substances that we are studying regarding the effect on the progression of the MASLD, and then a little bit of information data about the animal model that we are using and some preliminary results of the project. So first of all, a concept that you are all familiar about is the concept of MASLD. So this liver disease is characterized by different stages. So we go from an healthy liver, and then we start to accumulate fat. We go to a state of steatosis. Then when the inflammation kicks in, we progress towards a state of MASH. And then when the fibrosis comes, we pass to the status of cirrhosis. And the final stage is the hepatic cellular carcinoma stage. The only stage that are reversible are the steatotic ones, and the MASH. The others are not reversible. Another concept that is important to introduce here is the concept of exosome. So during our lifetime, we are exposed to different factors that can influence our health, can be related to lifestyles, like if we do exercise, what we eat, if we smoke and stuff. Then the ecosystem where we live, if we are more close to the nature, close to source of pollution, of viruses, bacteria. Then the social environment, like our circle of friends, the economical status, and finally, what is the chemicals that we are exposed to. The same is for the liver. So regarding the liver exposome that can influence the progression to MASLD, can be factors related to diet, infections, medical drugs, air pollution, how we behave. If we smoke, we assume of narcotics, of alcohol, and also the environmental occupational chemicals we are exposed to. So in our project, we are interested in the combined effect of the diet and the chemicals that we are exposed to, how these two factors can influence the progression towards the different stage of MASLD. So the chemicals that you can be exposed to are of different sources. So of course, it can be the outdoor and indoor air pollution, pesticide, fragrance product, flame retardants, persistent organic pollutants, plastics, food contaminants, and so on. So now, what is the EDC MASLD project I'm involved to, and the study the impact of these factors? It's a new project that started in January 2024, and is the EDC, it used the acronym EDC MASLD. It's an European-funded project that focuses on exploring the role of endocrine disrupting chemicals in the progression of metabolic dysfunction-associated steatotic liver disease, so MASLD. And the objective of this study is to characterize the impact of the environmental exposure to EDCs on the internal exposome, like metabolome, good microbiome, epigenome, proteome, immunome, and the degree of liver damage in MASLD in a perspective study setting. So this is just to present you the project. So this is a picture that we took at the annual meeting that we had last March in Turin, and this is the whole consortium. And this project is divided in different work packages, like VP1, VP2, that work package 1 and 2 that are more like the project coordinator, ethics requirement, and then the more scientific one are VP3, work package 3, that are related to integrative system biology, VP4, that is more on the clinical study side, meaning the impact of EDCs on humans, and then the VP5, what we are part of, is more related to in vivo and vitro models to study the mechanisms that these compounds affect in the MASLD progression. And then there are other more administrative related, like pathway to impact, activity and other dissemination work packages. So if you want to know more about this project, I put it here, the website, where you can have more information. So we have in this consortium, we are like 14 partners coming from different parts of Europe. And here is my group that is in Valencia, Centro de Universidad de Investigacion Principe Felipe. Then also, concerning this project, at the European level, we are part also of a cluster of projects with 4 more projects that study the endocrine disrupting chemicals at the level of other organs or system, like immune system, hypothalamus, pituitary axis, and so on. So, and this cluster is called Anchor. So coming back to the consortium, as I mentioned before, we are part of the work package 5 that entitled like mechanism of action of EDCs and their mixture in MASLD, and they are the leader is from the University of Utrecht. Our objective is to assess the EDC-related progression of MASLD in vivo models, and to determine which mechanisms are the most significant in EDC-related MASLD progression, and determine suitable marker for this mechanism for future screenings. Again, so there is some other concept that are, so what are the endocrine disrupting chemicals? Endocrine disrupting chemicals are defined as exogenous chemical or mixtures of chemicals that interfere with the endogenous hormonal signaling, and that affects the cellular metabolism. They are capable of mimicking or blocking the action of endogenous hormones, and a subset of these EDCs consists of what they are called obesogens, so that can promote obesity with towards different mechanisms. And then also in this kind of chemical, there are some that are called POPs, so persistent organic pollutants, so that can persist long periods of time in the environment and can be accumulate. So the more you are exposed, the more you accumulate in your body. And as you can see here, they are of very different nature and are being used in different, added to different products. So this is just to show you when the first time that this term endocrine disruptor was used. It was in 1991, and then from that date, there was the evolution of the studies and the research, with more and more people being more and more aware of how these compounds are dangerous and can affect our health. So now, again, among these endocrine disrupting chemicals, there is the per or polyfluoroalkyl substances, also known as PFAS. These PFAS came into use after the invention of the Teflon in 1938, and that was used to make fluoropolymer coating and products that resist heat, oil, stains, grease, and water. So you can find these PFAS in different objects that we use in our life, like dental floss, cleaning products, makeup, pesticide, water-resistant clothing, and so on. And these PFAS are part of the POPs, so they are very persistent in the environment for a long time. So why? Because these chemicals are a group of synthetic fluorine chemicals that have multiple fluorine atoms attached to the alkyl chain. So the fluorine replace the hydrogens, and that's what makes them very, very persistent, because these carbon fluorine bonds, they don't degrade easily. So that's they are called POPs, or forever chemicals. And of these PFAS, they are a long list. So there is more than 5,000 of them that have been generated. And for our study, we decided to focus on the perfluorooctanoic acid, called also P4. So here is just to show you that worldwide, this is a study in 2019, where some of the PFAS, like P4 and PFOS, can be found in the drinking water, and the supply water, drinking water supplies in different parts of the world. And in different parts of the world also, you can be exposed to more or less amount of them. For example, in the United States, the amount of exposure is very huge. With respect, for example, to Spain, where I am now, that is minimal. And here, just to give another example of another study, is a study done from 2014 to 2021, where they measured the level of PFAS in the blood, of different PFAS in the blood of teenagers in 9 European countries. And the levels of PFAS in the blood of these teenagers were above the health-based guidance value. So you can see that, for example, I mean, among the different European countries, there is different percentage. When the lower, like in Spain, like I mentioned before, or in France or in Sweden, where it's much higher. So depending where you live, you are more or less exposed to this kind of chemicals. So as part of the project EDC MASLD, we plan to test different compounds, but we have decided for the in vivo study to start from the P4. Why? Because it's one of the most common PFAS that have been used. It's classified as a long-chain fatty acid, PFAS, sorry. Was banned in the US in 2014 and in Europe in 2019, However, considering that its half-life is estimated to be 8 or 10 years in the body, it's still a matter of danger for our health. And also it can be found in the water that we drink. So the mechanism of actions that these PFAS are believed to act through to promote the progression toward MASLD is to interacting with some nuclear receptors like PPAR alpha or FXR of others that cause an alteration of their target and an alteration of the lipid metabolism. So our working hypothesis that the exposure to a moderate dose of P4 could induce significant changes in the liver function and morphology in vivo, promoting the development of MASLD. So now I wanted to give you some an overview of some of the studies that have been done on PFAS and P4 and why we decided to anyway go on with studying this compound. So this is a systematic review that has been published in 2022. Here they did a systematic review and a meta-analysis, and they evaluate the PFAS exposure effects on liver in human and murine models. So this study included 85 rodent study and 24 epidemiological work on humans. So the main results, in summary, of this systematic review was that the PFAS in general increased the liver weight. They cause an histopathological alteration of after exposure in the liver, mainly steatosis. This was mainly related to P4. Then the effect is more severe in male than females, and there is a systemic toxicity caused by this compound. Damage, meaning like damage in other organs. Then focusing more on the P4, what has been published so far about the effect on the liver? This is a study that has been done in 2013, where they used only males, and of the strain C57 Black 6N, they fed them for months on high-fat diet, and for the last 3 weeks, they treated them with P4 at a very high dose, like 5 milligrams per kilo per day. And what they saw is that the body weight of the mice were decreased, but, both in chow diet and in high-fat diet, but the liver weight as a hallmark of the exposure of this compound is increased. And also we have an increase on the levels of some of the biomarker of the markers of liver damage like ALT, AST, and ALP. What they saw at the level of the liver histopathology is that the P4 here caused this arrow here, also they are small, indicate some lipid droplets. So that correlates with the increase of triglyceride that they observed there in the mice here treated with P4, both in the chow and the control diet. And also they saw here these arrowheads, some infiltration of inflammatory cells. And they look also in this case at the epididymal white adipose tissue on these mice, and they saw that even though the adipocytes were smaller, what indicate a better health, there were a huge infiltration of immune cells here, like Crohn-like structure around the adipocyte. So as conclusion, they had that high-fat and P4 exposure synergistically induced liver injury, including lipid accumulation, inflammation, necrotic cell death. And I didn't show here, but they look also at the gene expression metabolomic analysis that showed that both alpha-DI and P4 significantly disturb hepatic metabolism in association with activation of PPAR alpha. So then here we have another study done in 2016 that in this case they use more both female and males from two different mouse strains, like C57 Black 6 and BALB/c. They used a dose of P4 that was much less, like 10 times less than the one used before. And they treated the mice with P4 and alpha diet, supplemented by cholesterol for 6 weeks. So here in this case, they didn't see any difference in the body weight, both in female and males. But what they observed, like a hallmark of the exposure of P4, an increase in the liver weight here. And then they look at the plasma cholesterol and at the liver cholesterol. And concerning the plasma cholesterol, they saw an increase in the treated mice, but to a different extent depending of the strain of the mice. And here a decrease specifically in the BALB/c regarding the level of cholesterol in livers. And then looking at the activation of PPAR alpha here, they saw that only they had a significant stimulation of this pathway in the BALB/c male mice. So, yes. In conclusion, so the C57 Black 6 and BALB/c male mice exposed to P4 present no change in body weight, increase in liver weight and plasma cholesterol levels to different extent depending on the mouse strain. And the BALB/c mice treated with P4 showed decreased liver cholesterol levels and PPAR alpha signaling induction. Now, the last study that I wanted to show you has been published in 2022, so more recently, and it's a more long-term study that is for 28 weeks, and it's only done, unfortunately, only on males in this case. And we, I'm talking about only this group, the control, the P4 treated with a lower dose, 0.05, and a highest dose, 0.3 milligrams kilogram per day. So I'm focusing only on these three groups. So what they saw in this, they treated the mice with P4 chronically for the whole 28 weeks. So what they observe here is that a lower dose of P4 that is here in the light green didn't change anything in the body weight, but the highest lows decreased the body weight of the animals. Considering the liver triglyceride, both the doses, the lowest and the highest, cause an increase in the weight of the mice, of the liver of the mice. You can please have a look only at the solid bars. Here you have an increase, and here also we have an increase in the highest dose, but to a lower level. And when they look at the fat mass and liver weight, the fat mass was not changed when the mice were exposed to a lower dose of P4, but they had a decrease in the body weight with the highest dose. And concerning the liver weight was the opposite way around, meaning that the lower dose didn't change much the weight of the liver, but the highest dose did. Then they performed some glucose and insulin tolerance tests on these mice. And what they observed is that there was no changes in the wild-type mice treated with P4 with the lowest dose, but they haven't improved in the glucose tolerance test with the highest of P4. And the same was for the insulin tolerance test. So that indicate that different doses of P4 cause different phenotypes. So 0.05 milligram per kilogram per day promote increase in liver triglyceride, but body weight, liver weight, fat mass, and glucose tolerance and insulin tolerance tests were very similar to the wild-type mice, meaning that could be explained with a local level in the liver caused by this dose. But the highest dose, the 0.3 milligram kilogram a day, promoted increase in liver weight, but lower body weight and fat mass, improved GDT and ITT, what can indicate a systemic effect of this dose. So consider, depending on the dose you are exposed, you could have a more local effect on the organ, or with another dose, a more systemic effect. And when they were looking at the transcriptome of the livers, you could see here that the more significant change in the gene expression was with the highest dose. It's like a little bit of a dose-dependent change. And if we look at the genes target, the PPAR alpha target genes, they were highly upregulated with P4 at 0.3. That means that indeed the P4 is targeting PPAR alpha pathway, but also if you look here in the mice that are PPAR alpha knockout, there is also an increase of expression of other genes, respect of controls, and this signature is similar to the one of the activation of PXR and CAR nuclear receptor. PCN and TCPOBOP are respectively an activator and of PXR and an activator of CAR. So that means that the metabolic and anti-transcriptional effect of P4 were mainly mediated by PPAR alpha, although the involvement of PXR and CAR was also evident. So, and another thing that is important to mention about the P4 is that it has been described to have a direct effect on the activation of the brown adipose tissue. So brown adipocytes that have been treated with P4 or P4 in this case, two different PFAS, they can cause decrease in food intake. There are some studies that show that, and some others that they didn't see any difference in food intake, but both of them, all of them show that they can have a direct effect in activating UCP1 and promote thermogenesis, and as a consequence, cause a decrease in the fat mass and body weight. So in conclusion, P4 can promote direct activation of UCP1 and thermogenesis and decrease fat mass and body weight, and it can act like a natural fatty acid to activate UCP1. So it could target direct UCP1 and have the same effect that an endogenous fatty acid. So considering that all the study that we have been done so far have been done in different mouse strains and also using diet, they were not really targeting the induction of MASLD that we have sometimes one gender, another gender, and not both of them in the same study. What we wanted to do now in our project is to do a comprehensive study using both genders and use a mouse model that specifically is the best representative of MASLD progression, and also using doses of P4 that are close to the human, to the one that at which the human are exposed. And on top of that, we want really also to have a look to the interaction between brown adipose tissue and liver in this model. So for our study, to select the mouse model, we had a look to this paper that was published in 2024 in Natural Metabolism, where they did an unbiased ranking of the murine dietary models for MASLD progression with respect to their similarity to the human disease. And also the diet to which the different mouse models were exposed was evaluated with respect to its ability to induce metabolic dysfunction and fibrosis. In this study, we were used more than 39 mouse models and more than 500 animals divided in controls and diet exposed mice. So the mouse model that we choose for our project was ranked here in the first, is the one highlighted in red, that was ranked in the first position for its ability to induce metabolic dysfunction and fibrosis, similarly to the human disease, using the human proximity score that was generated and developed in this paper that I will not go too much into detail, but so we selected this model for our study. And so going back to our working hypothesis, we decided to perform first a pilot study of 1 month and then a chronic exposure experiment from 9 months. So I will focus on on the pilot study. So why we needed to do this pilot study? First, because we wanted to verify whether the compound could create an aversion to consumption and modified with food intake and water intake. To check, in fact, if the water that we were using to dilute the compound provided to the animals contained traces of P4, considering that all of us, we are exposed to it. And assess whether this could alter the administered dose. And finally, to confirm that the compounds were effectively reaching the liver. So this was our experimental design. So we use, so the mouse model indicated by the 2024 Natural Metabolism paper was not only the diet was the GAN diet, the Gubra amylin diet, that we used only in the chronic experiment. And the chow diet is the Ultramin diet 1, 2, 3, 4, but also depend on the mouse strain. So the mouse strain chosen was the C57 Black 6 and taconic. So we took 4 males and 8 males and 8 females, divided by the 4 and 4 treated with the vehicle, and 4 and 4 treated with the high dose of P4 that we will use in the chronic experiment. We collected urine, feces, and serum at 2 times, at the beginning and at the end. And then we were weighting the different organs, taking the liver and weighting it, and also doing some metabolomic analysis. So here are some preliminary results that we got. So considering the liver size and weight, we saw an increase both in the size you can see here and in the weight that was significant more in the males. So it was significant in the males, but only a tendency in the females. And then when we looked at the morphology of the liver, this is representative both for the males and the female. What we saw is that there is an alteration in the liver morphology, showing a zone-specific increase in the size of the hepatocytes. Can you see here that are bigger than the one here? Then we perform some metabolomic analysis of the liver. And what we saw is that, first of all, they separate. So the principal component 1 separate the P4 treated mice from the control mice, and the PC2 separate the males from the females. So here you can see in the metabolome that there is clearly a different sex difference, what is expected, and that indeed the P4 and its derivative were detected in the liver of the P4 treated mice here and here. And there were some metabolites that were expressed, found overrepresented in the P4 livers, treated mice. The difference were more, how do you say, significant in the female than in the male. You can see here that it's not that clear. And then we performed also some lipidomic analysis. And again, that reveals some sex-dependent changes. You can clearly see here the difference by gender. And the P4 treatment caused alteration in lipid species, mainly in the male mice. You can see here that the changes are bigger in the males. With increasing level, significant increased level of LPE, LPC, PS, and carnitine indicated here. So then it's just the preliminary results of the pilot study. And then considering the chronic exposure, I can only show you the experimental design because we are starting it now, because I mean the project is very, very young, so it's only 1.5 years that we are doing that. So what we plan to do, or what we are progressing to do in the chronic experiment, is a really comprehensive one. So we will use the same mouse model and using the gubramylin gum diet to promote the development of MASLD. So we will have 6 groups. So for both genders, so we will have 48 males and 48 females. There will be groups of 8 mice for each sex exposed to Altromin diet and vehicle, to the low dose, and the same for the high dose. Same for the Gubra treated fed mice. Then the study will be of 38 weeks. I know that in the paper of in 2004 in Natural Metabolism was 42 weeks, but looking at the literature, there will not be much difference between a 38 and a 42 weeks, considering the length of the study. And during this whole period, we will collect blood, feces, and urine at different time points, more or less here indicated. And then we will do a glucose tolerance test, insulin tolerance test, and also indirect calorimetry, so to measure the oxygen consumption, to see if there is any, how do you say, the brown adipose tissue, any thermogenesis activation in these mice, as suggested from the literature. Then we will take the different organs, mainly focus on the liver, where for which we will do some RNA-seq analysis and repeat the metabolomic analysis that we did for the pilot. So, in conclusion, some preliminary conclusion only, is that P4 treatment in the pilot, the P4 treatment did not cause any adverse tasting. It was not detected at it was the traces of the compound in the drinking water and in the food, because we checked also the diet that we were giving to the mice, because we were afraid that maybe the plastic container where they will expose to before. We didn't find any trace of it, or very, very minimal, that will not affect the dose given to the mice. And this P4 and derivative were detected in the livers of the mice, what result in an increase in the liver size weight and zone-specific hepatocyte sites increase. And in the liver alteration we are mainly observing with respect to lipid species. And the most significant changes we're observing the male mice, indicating generally dysmorphism, which is characteristic of an EDC like the P4E. And in progress, of course, we are further analyzing the pilot study samples to increase our knowledge about the compound effect, and also progressing in the chronic exposure experiment. So that's all. So thank you very much for listening to me, and I'm open to questions if you have any.